So this is our second Tick Talk of the year for today, 11 March 2020. And while we are sitting on the precipice of another pandemic crisis that we all have in our minds, we are also coming up in tick season. So I wanted to talk about a few aspects of tick biology, in particular related to tick feeding and tick and associated behaviors, so that we could have a little better understanding of the risks associated with these organisms. Just a reminder of where we stand in terms of the upcoming risk. These are data from the Laboratory Medical Zoology's Tick Report program. You can always download these data yourself or search a narrower scope of them by your state or county or zip code by going to tickreport.com/stats, which you see at the bottom of the slide here. These data for 2019, these are submissions we've received for deer ticks, dog ticks, and Lone Star ticks. Predominantly human biting ticks. In some cases, people send us ticks that have fed on their pets, although that's a relatively small percentage. And what you see is that where we stand today is very close to what's going to be the coming adult deer tick season in the next couple of weeks, which will really kick off once we get into the month of April, about the 12th or 13th Julian week of the year. And you'll see here a couple of things. First, you'll see that there's a bimodal distribution to the activity of deer ticks, unimodal distribution of dog tick and Lone Star tick bites. You'll also notice that there's still substantially more deer tick bites than dog or Lone Star tick bites throughout the range where we're receiving ticks from. I would say that there are certain locations, for example, in Long Island, where we're receiving more ticks, more Lone Star ticks than deer ticks at this point. So it's a question as to whether there may be something going on with respect to diminishing numbers of deer ticks in those sites and an increase in Lone Star ticks. I just want to show you now those three different species. Here's the dog tick distribution. So I've just subtracted out now the other two species, and you see that unimodal distribution and a unimodal distribution of Lone Star ticks. This isn't as smooth, primarily because the numbers are smaller, and so the distribution is a little bit spottier. And then, of course, the bimodal distribution of the deer ticks coming in the spring, summer, and then recurring again in the fall. And if we look at that more closely, look just at the deer ticks, we see that what's comprised there is 2 activity cycles for the adult deer ticks and a single distribution of nymphal ticks, which won't start until sometime around May and then reach a peak in sometime in June into July. We emphasize that it's the deer ticks that are the principal reservoirs of Lyme disease, and that Lyme disease is the most common reported ailment associated with tick-borne diseases, with tick bites, but there's always risk from the other species as well. So that's just an update on where we stand with respect to the upcoming tick season. I wanted to spend a little bit of time today just talking about tick feeding behavior and associating anatomy and physiology. I want to point out that, as always, there are very good resources. I don't want to replicate things that have been done elsewhere by people like Larry Dapsis on Cape Cod. He has a wonderful series of videos that show different aspects of tick biology, including what he calls tick bitology. If you haven't gotten a chance, you can go onto our website, tickdiseases.org, to get web links to his video series. Also, Tick Encounter is always a great resource. And of course, Kirby Stafford's book, which is available for PDF, and you can download that on our website as well. So I'll try not to replicate what's covered on those other resources, but I will try to sort of supplement it with some additional information about, among other things, preference for hosts, behaviors to maximize host seeking while avoiding hazards, and then specialized body parts and structures that are used in the blood feeding process. So just a reminder that tick behaviors in general, or tick preferences, can be characterized by the number of hosts that they utilize to complete their life cycle. Many of the ticks that we're concerned about, including all of those 3 major species of human biting ticks, Ixodes scapularis, Dermacentor variabilis, and Amblyomma americanum, the black-legged tick, dog tick, and Lone Star tick, are 3 host ticks. This means that they feed on 3 different host organisms in the course of their life cycle as a larval, nymphal, and then adult ticks. In the case of Ixodes, that's maybe 3 different species of hosts, whereas in things like Amblyomma americanum, it may be a single species, but 3 different individuals. It's important to recognize that 3 host tick species references the number of individuals they feed on. I think that becomes more clear when we look at a 2-host tick. In the case of 2-host ticks, unlike the 3-host tick, they'll feed on just 2 individuals over 3 life stages. So the larvae and nymph will feed on a single animal. The larvae will feed, molt, and then continue to feed as nymphs on that same host. And then later they'll fall off and then later feed on a second host. In this case, a switch from a rabbit host to a cattle host. And then of course there are single host ticks. And the distinction here is that these ticks will remain on the host and feed larval, nymphal, and adult blood meals all on the same individual host. So the cattle tick Rhipicephalus annulatus has this life cycle. So too the moose ticks Dermacentor albopictus have a single host lifestyle. So you can imagine that for a single host lifestyle, while there is risk of transmission to that host, there's less spillover, which is the thing that most concerns us in terms of human health, spillover from one species to another when they choose to feed on different hosts at different stages. So the number of hosts shouldn't be confused with host preference. As I indicated before, that more speaks to the number of individuals they use to complete their life cycle. In terms of host preference, we can talk about generalist parasites or specialist parasites. And here these 2 scenarios sort of shape up. You can see on under the leftmost figure, in the instance when a tick has a broad and general choice of hosts, it will feed on multiple species and potentially acquire multiple pathogens from those different hosts and transmit them across host species barriers. This generally describes the black-legged ticks, the ones that are most of interest to us, and they pose risk because they're picking up pathogens from those different systems and then transmitting them to humans via spillover. Other hosts that are more specialized, for example, Lone Star ticks, would correspond to this. So this would be a scenario that would be reflective of the Lone Star tick, where the Lone Star ticks, while they change individuals, they feed predominantly on white-tailed deer. And so the pathogen transmission from them tends to be more specialized, reflect more of a specialized set of blood meals. And then in some cases, ectoparasites may be exclusively pathogens of avian species. Those species that tend to feed on rodents, either exclusively or as a major part of their generalist diet, are the ones that most commonly involve spillover pathogens into humans. And then, of course, reptiles can also serve as specialist hosts for certain species of ticks. But the nature of risk associated with tick bites will depend on these different types of host preferences and different alignments of pathogens or microbial parasites with the host of the blood meal. So what we're going to break down here is the behaviors that ticks use first to find their host, attach to their host, feed on those hosts, and then facilitate the blood feeding process. So this is, as we all know, the classic behavior of many species of ticks. It's a questing behavior. They crawl out to the edge of vegetation. And the point here to emphasize is that ticks don't jump, they don't fall from trees, they don't fly, they quest, and they wait for a host to come by, and then they jump on that host, and then they seek a place that's suitable for blood feeding. They insert their mouth parts, they cement themselves into place, and then undertake this lengthy process of blood feeding, which may involve transmission of pathogens, as shown here. But basically, they're secreting materials from their salivary glands, they're sucking blood from the site of the feeding lesion, and that's how they're imbibing their blood meal. And this obviously is of concern to us when it involves transmission of a pathogen that comes into us that may make us ill. So just a word about questing behavior in general. ecologists have done some great work by Tompkins here that I've cited, to look at how questing behavior can be correlated with temperatures. And what we find is that, or what they've suggested is that as temperatures increase, so there's a distribution of annual temperatures shown by that blue curve, that as the temperatures increase, we see an increase in questing behaviors, or an increase in propensity for questing, which corresponds to a decrease in resting behavior. And at some point, some temperature is indicated by that dark gray arrow, you would see a trade-off where the temperature sort of reaches a sweet spot such that the ticks will tend to quest more often than rest. And so what they're balancing here, the trade-off here, is the need to find that next blood meal, trade-off with the need for basically avoiding desiccation conditions and or predation. So they want to take the risk of going up to the end of the branch where they can be likely to find a host, which they won't find when they're deep in the leaf litter, but that comes with costs associated with desiccation due to higher temperatures and drier conditions, and also makes them susceptible to predation. So ecologists have tried to quantitate this and look at the evolutionary aspects so that we can better understand how changes can lead to adaptation. For example, if you imagined a shift in the temperature distribution, upwards or downwards, as could be associated with climate change or movement or migration to new habitats, then you can imagine that this resting questing trade-off could shift in a way that the critical point could increase, so that perhaps ticks can adapt to questing at higher or lower temperatures in response to the environmental conditions. Other people have looked at other factors of this trade-off, including energy budget trade-offs. So it costs energy for a tick to do that business of crawling up to the top of the branch to go on a quest. And given that ticks feed once in each of their life stages, so once as a larva, once as a nymph, and once as an adult, it means that they may go many, many months without imbibing any further nutrition. So they've got to make the most out of the nutrition that they got from that previous blood meal. And one of the trade-offs that they would use is how they're going to use those imbibed carbohydrates to determine when and if they'll get that next blood meal. So in addition to temperature and climate responses, there are probably adaptations that have to do with modifying the behavior to maximize energy budgets. So where and when ticks will quest is a factor that's determined by those different environmental components. And then once they decide that they're going to quest, they have certain sensory apparati. So ticks can smell. They smell with organs called the Haller's organ, which are located on the end of their legs or feet, if you will. So the Haller's organ is a sense organ that can detect, among other things, CO2 or pheromones given off by hosts and help them to locate hosts. Different ticks rely on their sense of smell and their sense of detection of hosts to find those hosts, depending on whether they tend to be more ambush predators that lie completely in wait, as deer ticks do, or whether they take a more active role in seeking out their host, as lone star ticks do. So lone star ticks generally will respond to those olfactory cues and crawl towards a host, whereas deer ticks tend to stay put and wait for things to come by. But both of them have Haller's organs that they use to sense and determine the difference between a host and an environmental background. So just a word about anatomy. Ticks differ in size and shape. We know that. Here's the three common species of human-biting ticks found in the northeastern United States: the deer tick, the dog tick, and the lone star tick. They differ not only between species in terms of their anatomy, but also between individual life stages. You can see there's major size differences between the juvenile stages. In each of these four species, I've given you the larval, the nymphal, and the adult male and female representations of each species. And if you look across species, you can see the difference in for example, the anatomical mouth parts. Some have very long, elongated mouth parts, some have shorter, stubbier mouth parts. And that reflects different feeding strategies, as we'll see in a second, that will determine how and when the tick feeds, how it uses cement and other things to secure itself into place. This is also a good point to remind you. So I'm borrowing this or lifting this directly from Tick Encounter. When it comes to identifying the different tick species, I just want to remind people that while these images show very clearly the distinction between deer ticks on the far left, dog ticks in the middle, and Lone Star ticks on the right, once a tick has become fed, as shown in this series from the Tick Encounter resource pages, this tick that looks much less like its fully fed counterpart than we would like, despite the fact that all 6 of these along the top are the same species. And Tom Mather and his group at Tick Encounter have pointed out that there are aspects of the tick anatomy that don't change through the course of that feeding, and in particular, different anatomical features on the dorsal side of the tick remain constant and still serve as the identifying features that distinguish between species. So even though a tick has moved from here to here and has lost this orange margin on its body, the capitulin parts still remain the same in terms of pigmentation. So you can go on Tick Encounter and review that information on how to identify ticks. The other thing to remind you of is that the principal concern here is in this tick, which is the nymphal stage of the deer tick, which won't be active for another couple of months here. And the thinking is because these ticks are so small and because they've taken at least one blood meal and have the potential to be infected, on average about 30% of them have Borrelia burgdorferi, the causative agent of Lyme disease, they pose some of the most serious health risks because being smaller, they tend to feed longer, and the longer a tick feeds, the more likely it is to transmit pathogens if it should have them. Just rounding out our conversation about a tick anatomy, so here's a cutaway image of a tick showing its internal anatomy, nondescript species here. There's a couple of organs that are of particular interest to us inside. We're going to spend some time talking about these things, the salivary glands. Notice the salivary glands make up a major portion of the internal tick anatomy. They serve a number of important roles in feeding and other aspects of tick biology. So too the midgut, shown here in red, because eventually it will be filled up with blood, which is the sole source of nutrition for ticks. It's connected to the outside world via the hypostome, which is this hollow barbed tube at the front end of the tick, where the blood flows through, and I'll show you some really neat anatomy of this in a moment. Ticks have hemocoel, so they have an open circulatory system. They don't have blood in the context that we think of it, although they do have their own version of that, and that constitutes the hemocoel, that open circulatory system in the tick. And I should mention that the hemocoel, this is how the tick protects itself from infection. So like us, ticks need to avoid infection with harmful bacteria, fungi, parasites, and otherwise. And so the hemocoel is actually a pretty hostile environment to the microbial world. There are antimicrobial peptides like defensins and other molecules circulating in that open circulatory system and tend to have high activity against microbes. And so microbes that, like Lyme disease, that persist in ticks have overcome the ability of the tick to kill them. So they're really finely tuned and well adapted and haven't just appeared from nowhere. They actually have had to co-adapt with their host vectors, with their tick vectors in order to survive that antimicrobial habitat. The other thing to understand about remember about an open circulatory system is that ticks don't have lungs per se. They don't do an active gas exchange where they draw oxygen into their body to oxygenate that hemocoel. The gas exchange takes place through the spiracles, which are located on the outside of the ticks. And basically, you can think of these as tunnels that allow the gas to come in and exchange with that within the hemocoel and facilitate the oxygenation. So it's a more of a passive gas exchange, and it's the means by which ticks basically breathe. So I emphasize that because one of the things you'll see commonly, one of the myths that's commonly put out there is that you can remove a tick by adding petroleum jelly or nail polish, and that will suffocate the tick. Because of the passive way that ticks ostensibly breathe, it takes a long time to suffocate them in that manner. And as we'll show you in a moment, it's very important to get ticks off as quickly as you can. So hoping for a long-term suffocation is much less preferred to just removing the tick as quickly as you can. This image shows what the spiracles look like, those openings that allow the gas exchange, and very often they're used for taxonomy because they do differ from species to species in terms of the shape and the types of punctation they have. The other two features I want to point out about external anatomy of ticks are the chelicera, shown here at the bottom, and the hypostome. So the hypostome we talked about before, and people are probably familiar, you've probably seen pictures of the hypostome. In this image, actually, the hypostome is shown underneath these chelicera. The hypostome is the barbs are sticking out here on the end. That's the structure, that's the hollow tube through which the blood will be imbibed when the tick starts feeding. They are barbed on the outside so that they secure the tick in place. The chelicerae are the teeth. And when we talk about tick feeding, we should not think of chewing because the teeth of the tick are not chewing teeth, but more clawing teeth. In fact, the name chelicera derives from the word for claw. As ticks feed, they sort of claw their way into the tissue. They claw their way into the host that they're feeding upon, and they do so using these chelicerae, which work back and forth kind of in a sawing motion to cut their way, if you will, into the site of the wound. We should also mention the palps, which are the other two features at the anatomical anterior end of the tick. The palps are used to secrete the cementum, the adhesion that glues the tick into place. So between the cement and the barbed hypostome, ticks are pretty securely in place when they're fully feeding. There's a video showing the tick feeding apparatus, the chelicerae. Just to orient you, this is the dorsal side of the tick here. This is the ventral side. These are the mouth parts that are sticking into a glass capillary. This is a really great photography work by Vankova et al. And what I'm going to show you when we activate the video is the sawing motion of the chelicerae cutting back and forth, and in this case, not cutting into anything other than the liquid substrate they're exposed to. That's the chelicerae clawing at the tissue, so they would be tearing the tissue with each one of those claw strokes and pulling that hypostome, the thing down below the chelicerae, into the site of the feeding lesion. And that's basically going to be the straw that's going to pull the blood out of that feeding lesion. And there's just that was from the side. This is the top angle, again, showing that clawing action. This is what happens when a tick finds its way onto you and finds the place where it wants to take the blood meal on you. It uses these mouth parts to claw its way in. So these are cross sections showing the different types of mouth parts. So remember I said that the anatomical anterior end differs between species. This sort of gives you a picture of how different adaptations of those mouth parts have evolved and how they use, in some cases, a longer hypostome, as shown here, and actually cement themselves laterally to that hypostome, as opposed to some of the shorter, stubbier mouth parts where they may not reach quite so far into the dermis of the host they're feeding on, and use more of the adhesion. Adhesion is shown in the yellow here to facilitate their adherence to that tick. So different strategies for making sure that the tick attaches to and maintains its fixture on that host, because it's going to stay on there. In the case of adult ticks, they can stay on for as long as 2 weeks to complete their blood meal. And there are some species, as you can see, since it's biology, there are always some exceptions to the rule. There are some species that rely almost exclusively on the hypostome and not the adhesins to keep themselves in place and secured in that biting wound. So not surprisingly, as we forecast when we looked at the first internal anatomy of the ticks, salivary glands figure prominently in the feeding role. Saliva is an important component of blood feeding, and the salivary glands produce a number of different components that make the feeding possible and other behaviors possible as well. I'm not sure how well this picture shows up, but this is a cutaway image of an actual tick dissected. So here's the tick with its external carapace in place. Here it is cut away, and you can see this fluffy material that looks almost like cotton. These are salivary glands. Salivary glands constitute a major portion of the tick's body. And within those salivary glands, if we blow them up, we see that there are these little acini, little glands, and there are different types of acini that produce different active components of the saliva. There are dozens, scores, if not hundreds of different components of the saliva, many of which we don't fully understand what their function is, but there is major efforts to dig in and try to understand those better, because understanding that this is an important aspect of tick biology corresponds to also potential for finding areas to intervene in the feeding cycle. So tick saliva is multi-talented, we'd say. So it facilitates feeding. It does things like numbing the bite site so that the host that's going to be serving as a host for sometimes up to 2 weeks doesn't feel irritated in a way that it has to scratch that off. Now, there are consequences of this. Sometimes host organisms, including ourselves, will develop allergies to some of those components, and so that can be a protection that hosts have against feeding. Nonetheless, there are active components of that are meant to numb the bite site. They also have to facilitate movement of the blood through that structure. And interestingly enough, there are components of the blood system that probably suppress the immune response. So that's the vertebrate immune response, or in our case, the human immune response. Our bodies are going to respond to having this organism attached to us and spitting saliva into us. And so they have adapted through millions of years of evolution mechanisms that suppress that immune system. In particular, aspects of the innate immune response, things such as the complement system, they've developed mechanisms that allow them to block those cascading events that would otherwise interfere with the blood feeding process. But there are other aspects that aren't necessarily related directly to feeding. For example, some of those acini, some of those specialized structures, actually can produce hygroscopic molecules that can be spit out to the end of the mouth parts during the time when the tick is not feeding. And because they're hygroscopic, they'll draw moisture from the air. And by this means, the ticks can rehydrate themselves by drawing environmental moisture through their hypostome. And that's all facilitated by these salivary compounds. We also know that there are antifreeze proteins that are produced in the saliva that facilitate cold weather adaptations and avoidance of hypothermic death. And it may be the case as well that in some cases these facilitation of these different antifreeze proteins can come about as a result of infection. For example, anaplasma acquisition seems to lead to the upregulation of antifreeze proteins, which have the collateral effect of creating biofilms inside the tick, which can facilitate infection with various types of microbial species. So saliva, as shown here, is and salivary glands are very important anatomical features of the tick midgut. I want to show you a video of the feeding process. So this is a cartoon, but it's based on some X-ray microscopy that they put together to show the tick feeding process. So here's a tick that's embedded. Its mouth parts are inserted into the wound. There's the chelicerae and the hypostome, and a structure called the salivarium, where the saliva are going to be communicated between the tick and the feeding site. And then you notice that all of the salivary components above here were in green, whereas all of the components that lead to the midgut are in red. And the emphasis here is that there's bidirectionality to the movement of things in and out of ticks, that the things coming out of the tick, the host would be on this end, at the other end of the hypostome, are coming out of the tick through the salivary glands. The saliva is the stuff that's being excreted out of the tick into the bite site. And the blood is moving in one direction, not into the salivary glands, but into the midgut, these structures of the pharynx and esophagus. And the two of them are not connected in any way, that the salivary glands, components of the salivary gland and the esophagus are not intermixing. It's important to bear that in mind so that we understand how transmission of pathogen takes place through ticks. So there's the chelicerae in action. The tick is sawing its way into place. It's inserted its mouth parts, and now it's secreting salivary compounds, which are facilitating those things we talked about before, numbing the bite site, suppressing the expression, and it has musculature that facilitates that movement. At some point, probably at least 24, if not 48 hours, it starts the process of bringing blood back from that feeding site. So again, there's muscles on the side here that are facilitating this process through the pharynx and into the esophagus, and then eventually, not shown here, but further left on this image, into the midgut. The movement of things from host into tick and from tick into host are very separate channels, very separate channels in terms of the separation of the esophagus pharynx from the salivary glands and the salivarium. So I thought that's a wonderful picture that's based on real anatomy, and it shows you firsthand how those different structures work together. I'd just like to return for a moment to a little bit more cartoon image of it so that we can emphasize the point to be made about pathogen transmission. So here is the same image I showed you before with the chelicerae and the hypostome. This tick is attached. Its feeding tube hypostome is in place. Its chelicerae have chewed its way in, and the palps have secreted the cement to secure the tick in place. Let's look at what's happening on the internal anatomy of that tick by taking a cutaway view. Here's my rendition of the midgut protein, the midgut structures. These would be bilateral. There would be, not just on a single side of the tick. I'm just showing you half because I want to show on the other half the bilateral salivary glands. And what I want to emphasize is that these two structures are separated by multiple membranes. So there's the membrane on the outside of the midgut, there's the membrane on the outside of the salivary glands, and then in between is that hemocoel, that open circulatory system that is rich with antimicrobial peptides. So the feeding process takes place like this. The blood moves into the midgut, where it will be digested within the midgut over the course of the weeks and months after the tick has fallen off its host. So this is re-emphasizing that notion that blood, stuff moving from the host into the tick, moves in one direction and is maintained in one set of structures. Things moving out of the tick via the salivary glands move by a very different means. And so when we start to consider a pathogen, as evidenced by the little star-shaped object here, these do flow through the hypostome, but are not at all associated with the salivary glands. They move through the hypostome directly in the midgut where they're sequestered. And in the case of Borrelia burgdorferi, the Lyme disease spirochete, these actually stay in the midgut throughout the feeding on that host from which they become infected. After the tick has fallen off, digests that blood meal, and many months later has molted, the pathogen remains in the midgut and really doesn't make the migration from the midgut to the salivary glands until the next time the tick feeds. So let me say that very carefully. The movement of the pathogen from the host into the tick takes place in host 1, and the movement of the pathogen from the midgut to the salivary glands only takes place on the next feeding in host number 2. Okay, so if the tick acquired an infection from a mouse as a larval stage, when it goes then to feed on a person in the nymphal stage, then and only then, when it has begun that feeding process as a nymph on the human, can the pathogen move from the midgut into the salivary glands. And the emphasis here is that this process takes many, many hours of feeding. It takes transformation of expression of tick proteins. It takes transformation in the expression of the Borrelia proteins. And it's not simply that the tick is functioning as a syringe that sucks and spits blood out, because in fact, the syringe that sucks is separated from the syringe that excretes, if you want to stick with that analogy of a syringe. And it's only through some real important physiological changes that the spirochetes can make their way, or the pathogens can make their way across, including traversing this open circulatory system, which is a very hostile environment. In the case of Lyme disease spirochetes, which we understand the best, we know that there are, in particular, proteins on the surface of the bacteria called OSPC, and that OSPC proteins have ligands that do things. They bind aspects, for example, of tick salivary proteins. They do things like interact with complement cascade proteins, components of the innate immune response, like C4b. They also interact with factors that are involved with clotting mechanisms. And clearly part of that is to stop the clotting so that the blood can flow through. But they also do a very interesting trick. Now we're talking about spirochetes, Borrelia burgdorferi, the spirochetes associated with Lyme disease. They have an outer surface protein that actually binds a plasminogen molecule and converts it to the active version of that molecule, plasmid, that comes from the vertebrate host. And here's why. Inside a tick, Borrelia burgdorferi has to make its way from the midgut through a membrane, as shown by this dark layer, into the hemocoel, and then through another layer of membrane, which is shown light, doesn't show well in this image, into the salivary glands. And to move across those membranes involves the action of enzymes called proteases. And lo and behold, the bacteria that cause Lyme disease don't produce their own proteases. They don't have the ability to make this migration that they need to make across these membranes. So they have to borrow the activity from other organisms. And what they do is they utilize the proteases from the host they just fed on, in particular, plasmin. They use that as the protease that's going to allow them to traverse these different molecules. So those outer surface molecules on the bacteria bind host plasminogen, convert it to the active form plasmin, and then basically use that as a saw to cut their way through these membranes from one organism or one organ inside the tick to another organ inside the tick. That's the reason that they need that blood meal in order for the spirochetes to move between the different organs. And that's also the reason that there's a delay between movement from the midgut to the salivary glands. And that's the reason that we generally think that it takes 24 to 48 hours on average for the migration to begin to happen, for a heavy migration of spirochetes from that midgut into the salivary glands. Now, some people will push back on that and say that it can happen in a lesser time frame, and that may be true. We are emphasizing that the 24 to 48 hours represents a distribution, and that there may be some spirochetes that come across before that, or some spirochetes that come across after that. But generally, this whole process takes time involving upregulation of these different proteins. So I guess to sort of wrap this up a little bit, I just emphasize that there are these very specialized structures, including the midgut, the salivary glands, that contribute, as well as the external anatomy, that contribute to the development of this feeding lesion. And it is truly a lesion, they're not cannulating vessels the way mosquitoes do. They're actually creating a small pool of blood that they'll drink from. And they'll use that blood to basically imbibe their blood meal in the final stages of their feeding process. This image I took from this paper by Lees and Bowman, and it was basically looking at neurobiology of the tick, and that's why they've emphasized these different. This is basically the brain of the tick, and I'll just mention that these are the sites, or these are the targets of many of the acaricides, the compounds used to kill ticks. Maybe we could talk about that in a separate tick talk. In closing, I'll say that the reason we think it's so important to understand this biology, and I've gone into a little bit more detail than may be useful to a lot of us, but I wanted to emphasize that this is a real biological process, and that it's fundamentally important to understand it, because we think that it's not just before the tick bites that we should be concerned about. We should be concerned, or we should be knowledgeable about what happens when the tick bites and what happens after the tick bite, because each of these are opportunities to try to mitigate the risk of associated disease. So even if you've failed, or even if your efforts were not 100% successful in keeping ticks off you, and or keeping ticks from biting you, there are things that you can still do to mitigate risk by either terminating the tick bite before it reaches a point of highest risk, and or looking at what's happened even after the tick is fully fed in order to understand that risk. I've shown this image before, it bears talking about again. That's because we really want to be careful in how we identify hazard and risk. I use this example that if we wanted to talk about the hazard associated with shark bites, we understand that the hazards are associated with human biting sharks or human feeding sharks. And the risk is just the quantitation of the number of shark bites that take place. And so while there might be lots of sharks out there ready to chomp on us, there are a relatively small number of cases that actually take of shark attack that actually occur. And that's because it's mitigated by exposure. And very often when we're talking about risk, we may be talking about this component, or we may be talking about this component, but we overlook sometimes the importance of the most critical aspect, which is exposure. This is as true in shark bites as it is for tick bites, that we often think about the hazard, which is the number of infected ticks that might be occurring in the wild. That information is not without value, but it doesn't tell the whole story. So too, and we often look to the reported incidence of human disease, although that doesn't really reflect necessarily the exposure of these ticks to people. It could be that it takes lots and lots of tick bites before we get the actual manifestations of disease. So again, the emphasis on exposure and the importance of the work that we're doing is to look at those exposure factors, which are understanding the tick species that are involved with tick bites, looking at the duration of feeding. That's a critical component emphasized by everyone that understands these tick bites. And of course, looking at the infection status, because we know not all ticks are infected with all the pathogens. And so it's important, or an important consideration, to know whether the tick that bit you has been infected. So to summarize today's talk, I'd just like to say that ticks have specialized behaviors, anatomy, and physiology that facilitate their blood-feeding lifestyle. We've touched upon a few of those different specialized features. The feeding adaptation, that these feeding adaptations facilitate pathogen transmission, because not illogically, the pathogens have co-evolved to take advantage of this feeding behavior, and they recognize that this intimate association between tick and host presents an opportunity for their transmission between those hosts. And then the last point that we made was that interrupting feeding can reduce exposure risk. So the earlier you can pull a tick off from completing its feeding cycle, the better likelihood you are of having a favorable outcome from that tick bite, or avoiding the risks of serious illness. As always, we remain here ready to answer your questions at the Laboratory of Medical Zoology. Reach out to us by email at info@tickreport.com. Go online at www.tickreport.com, and you can access our chat line there and speak to someone in real time if you have questions about ticks or tick-borne diseases, and direct any of your questions that you have about the webinar through those websites.